so far. I'm recording, I'm presenting. So let's take a look at the material we had talked about. We had introduced last time, but it's a great place to start. The karyotype shows the chromosomes that are present within an individual. This is showing them in condensed form as you would see them in the metaphase of mitosis. And the banding patterns are actually showing you region by region changes in the chemistry as those chromosomes fold up. You can see from the lower right-hand corner of this karyotype that this is an XY individual and therefore a male. As you survey the karyotype from top to bottom, we notice that every chromosome has its homologue. So at each number, chromosome number 1 definitely shows 2 homologous chromosomes, and that is true for every pair from 1 through 22. So adding in the X and Y, that's a total of 46, and so this is a normal human chromosome complement. It also means something else. This homology, pairs, one of these chromosomes comes from the father and one from the mother. Meiosis will make gametes in this male. They will be sperm that will combine one each of these chromosomes into a sperm that carries 23. When the egg is formed with a complete half set, so it has 23, fertilization will produce, again, in the fertilized egg, a karyotype just like this, unless it could be XX instead of XY. But that balance is very important in humans. It turns out that chromosome variation in some organisms is abundant. Plants vary their chromosome numbers all the time. You will do a chromosome count on a plant and see an extra chromosome. I discovered a plant in Arkansas that was a triploid instead of a diploid. So whereas it looked like the species that had 24 chromosomes and paired as 12 pairs, this was a triploid and it paired as 12 triplets and was almost sterile with respect to pollen production. So plants vary this all the time. In humans, there are only a few chromosomes where we see a live birth if the chromosome number is not 46. The XY pair is the one that seems to be the most tolerant of chromosome variation, and you can kind of see why. These are already different. The X and Y in a male already are not completely identical end to end, and therefore tolerating that variation is perhaps something that's accommodated by a special mechanism. So we do see XXX, we see XX, the normal, we see XO, a 45 number, in the female chromosome complements. In the male, in addition to XY, we see XXY, which presents as a female, but has in heightened levels of androgens. And so muscular development, bone development are somewhat different. And we also see XYY, which present as males, but other than that, their only notable feature is the sterility of their sperm. So this pair does allow some variation. The most common chromosome variation sits right here at 21, and that is a total chromosome number of 47 from having 3 number 21s that is tolerated at low frequency in human births and is called Down's syndrome. It is our most numerous, our most frequent chromosomal anomaly. So the picture I'm trying to paint here with the chromosomes, the organelles that hold the DNA The DNA holds all the genes that make a human being. And the rule for humans, but also for animals in general, is that the balance of this set, the balance of meiosis, producing regular gametes with 23 chromosomes, and fertilization, reconstituting this number of 46 with exact correspondence of diploid pairing, is an important feature in the fertility of human beings. Major patterns of inheritance vary in humans because we have these two different kinds of chromosomes. 1 through 22 have a homologous pair, one from dad, one from mom. When we have simple inheritance, meaning that the gene involved, shown here by colored balls, the gene involved is one that affects a single trait. One gene and these two alleles determine the trait. Then it's easier to see the effect than if 2, 3, 4, 5, 6 genes affect the trait. About 80% of your genotype falls within this simple inheritance category, determined by homologous pairs of the gene, 2 copies called alleles. Polygenic inheritance, there's a lot of features that we would see as a trait, like hair color, like height, eye color, skin color, all of those pigmentations that are affected not by 2 alleles at one gene, but 2, 3, 4 genes carrying 4, 6, 8 alleles, and then the pattern of variation is, it still may range, for example, from dark skin to light skin, but it's very difficult to do controlled crosses and actually come up with genotypes. So we're going to look at some examples of this autosomal inheritance, and the autosomes are those chromosomes that have a homologue to pair with. We see a different pattern of inheritance here in the sex chromosomes, X and Y. This is showing the effect that we call sex linkage, or more accurately, X linkage. The presence of the gene and the allele on the X chromosome would have an autosomal pattern in females because the females have 2 X's. But in males, as you can see here, the X chromosome with its 1 allele determines the trait. So if it's a recessive allele, which normally would take 2 copies, if yellow is recessive over here, you need yellow, yellow to get the yellow trait. Over here, 1 yellow would produce the recessive trait in males. So those patterned traits that we more frequently associate with males, things like color blindness, and especially red-green color blindness, is present on the X chromosome. Some forms of hemophilia are X-linked. And other male traits like male pattern baldness, although our classical example of male pattern baldness, it turns out, is affected by other alleles in some limited conditions. Inheritance involving autosomal chromosomes, that first example. We see now, since we have a combination of alleles, if we have a chromosome, 2 chromosomes, and each carry the red, then you get the trait for red. If you have 2 chromosomes each with yellow, you get the trait for yellow. And they are different. The question is, what happens when you have an individual who carries 1 red and 1 yellow? Now the interaction of these 2 alleles determine the trait that is inherited. So the first example, and the simplest one to examine, is the one that Gregor Mendel selected as his example. This is how he came up with his explanation of factors, as he called them, that determined traits. He found 7 traits in pea plants, things like flower color, cotyledon color, that were exhibiting a consistent pattern of inheritance. So he selected and studied those. He came up with a hypothetical explanation that said, well, these must be genes. These must be factors that occur in pairs because they're showing proportions of combination that reflect a not one factor inheritance, but 2. So he did his crosses in a specific way. Those crosses were always performed with pure breeding lines. So he would get a pure breeding line of peas that had purple flowers, meaning if he left them alone over here, they always produce purple flowers. On the other side of his garden, he had a group of plants that always produced white, no matter what he did. The first thing he did then was purify the factors, whatever they were, for purple and for white, and he separated. Then he crossed them, and a strange thing happens. When you cross purple with white, the seeds that produce that generation of offspring, called the F1 generation, the first hybrid generation, has all purple flowers. White completely disappeared. If you take those F1 individuals, which are all purple, and cross them to each other, you get a next generation in which you have purple and white flowers, but always in a specific ratio of 3/4 to 1/4. Now, Mendel came up with a terminology. He said, in this cross, the purple trait is dominant to the white trait because white disappears when both are present. Purple dominates. But the white factor is still present. And if you allow the white factors to combine with one another in pairs, they produce 1/4 of the offspring in the second generation, what would be the grandchildren, with white flowers. So strict dominance in humans is observed in a number of traits. And here the important thing is to compare the dominant trait with its recessive counterpart. They always occur in pairs. So normal skin pigmentation varies dramatically over the surface of the globe from translucent white, so lacking in pigment that you often will see a pinkish tinge to the skin from the blood vessels that are present, all the way to skin that is so darkly pigmented that it looks close to black, or even I've seen skin pigments from Africa and from India that seem to give off almost a blue sheen. They are so dark. So those would be the range of normal skin pigmentation. But in all of those societies, regardless of what normal is, there is a property called albinism. Individuals who have that very, very white skin and appear pink because of the capillaries in their skin. They also have red eyes. There is no pigment in their irises. And a true albino is recessive to the normal skin pigmentation of the population where it arose. Freckles is another dominant trait. Nearsightedness and farsightedness are both dominant to normal vision, which is recessive. Astigmatism, it is another dominant trait. Notice the dominant traits that affect eyesight. Free earlobes is dominant to attached. Tongue rolling is dominant to the inability to roll your tongue. That's side to side. The Rh factor, Rh plus, is dominant to Rh negative. Type A is dominant to type O, and type B is dominant to type O. Type O is recessive to both of those other alleles. Huntington's disease is a blood disease that is dominant. A neural disease. Cystic fibrosis, Tay-Sachs, and phenylketonuria are examples of inborn errors of metabolism, genetic diseases that are recessive. But they all show this homologous diploid type of inheritance. Now, that's because of the expression of the proteins that are made by these two different genes. There are other genes that show codominance, which means that in the heterozygote, you don't see the disappearance of one type. You see a new type that is not like either of the homozygotes. So type AB blood is one that I'll mention. AA and AO, both are type A blood. That means A is dominant to O. BB and BO are both type B blood. That means B is dominant to O. But AB gives you a new blood type, type AB. Also, we notice a couple of biochemical traits. The blood proteins we called albumins and some of the transferins all show this codominant effect. Incomplete dominance is when two different alleles produce intermediate traits in the phenotype. So hemoglobin A production and hemoglobin S production. So in hemoglobin S, when you have, this is a mutation of one of the blood alleles, and the heterozygote for sickle cell produces a viable offspring, whereas the homozygote does not. Here's the way we analyzed these traits, and I'm sure you're remembering some of these. These are called Punnett squares. We do them in general biology. And in fact, the examination of human traits is, of course, of greatest interest to us in genetics. The idea that is shown here is you are crossing a mother, showing what happens when you have the recessive trait in the mother and you have homologs. So both the eggs carry little a, the recessive allele. The father is showing the dominant trait. So if the father is homozygous, all the sperm will carry big A. So you can see that each of these two types, and by the way, why do we draw two? It's because we have two chromosomes that are carrying this trait. So if you get the chromosome from dad's mother, you get the big A. If you get the chromosome from dad's father, you get the big A. But you can see then if you do each fertilization, each genotype is big A, little a. So all of these offspring are going to show the normal skin pigmentation. This is an example where father has normal skin pigmentation, no matter the race or location, and mother is an albino. So what happens when they reproduce? The only possible genotype in their children would be big A, little a, and all would show normal skin because the big A allele in this case is dominant. Now, I want you to notice one other thing. Despite the fact that each child has normal skin, each child will also be a carrier of the albinism gene. So that is the circumstance where a normal pigmentation person, big A, little a, if they choose as their mate another person with normal skin pigment, but who happens to be big A, little a as well, then the possibility of a little a offspring arises, and two normally pigmented people can produce an albino child. That will only be true for the recessive trait. The dominant trait will not operate that way. So let's take a look. Here is a maternal albino, a mother who's albino, and a father who has normal pigmentation but is a heterozygote, big A, little a. Now that means one of the chromosomes he received from one of his parents had a big A, and the other chromosome in that pair had the little a. Now you can see what is called a back cross. If you take the heterozygote and cross it to the recessive homozygote, you get half of the offspring are expected to have big A and little a and have normal pigmentation, whereas half of the offspring will have little a, and show albinism, in this case, the recessive trait. This 1-to-1 ratio is very easy to pick out in limited crosses and is considered a test for this type of inheritance. So what are we really talking about? One gene, we'll just call it the A gene, it affects skin pigmentation. Two alleles, big A for normal skin pigmentation, which is dominant, and little a for recessive skin pigmentation, albinism, which is totally recessive. Now, we have a number of different things we can say about sex, but the big thing we say about sex and sexual reproduction, our only method of reproduction, is that its big advantage is generating variety. Vegetative reproduction, clonal reproduction, generates exact genetic copies because it only requires one individual or even one section of an individual, one tissue. It is typically very rapid. You can basically take an appropriate plant and take cuttings from the tips of each branch, and the tip of each branch can immediately root and become a independently growing individual. So in terms of rapid reproduction, vegetative reproduction serves a lot of purposes. But everybody's the same genotype. And so in a variable environment where there's short-term and long-term climate change, when there's short and long-term weather patterns, that's a kind of a vulnerable position to have everyone have the same combination of genes. What sexual reproduction does in humans is it requires two parents, so two individuals, but it also requires this process called meiosis. And meiosis in a diploid organism produces huge and various arrays, combinations of genes. Here's one of the factors that contributes to that. The chromosomes, which are duplicated on the left here, in mitosis, what will happen is one copy will go right, left, and one copy will go right. While over here, the other one is lining up separately. So one copy will go left and one copy will go right. And there is an exact distribution in mitosis of these two purple and blue chromosomes. But in meiosis, they pair. Now all the strands of this chromosome number, let's say this is chromosome number 2, they all pair up. And while they're pairing, they show a phenomenon shown here called crossing over. Do you see how the light blue and the purple arms are crossing over? You can see this in the light microscope. In fact, when you look at meiosis, you often will count the number of crossovers per tetrad. So this might cross over, this would be 1. If this arm were also crossed over with this arm, and this arm will crossed over with this arm, that would be 3. And you calculate what's called a crossover frequency. Some species with smaller chromosome numbers have long chromosomes, and so these arms can twist around. You might, in each tetrad, you might see 3, 4, or 5 crossovers. So what does a crossover in the microscope mean? What actually happens is that enzymes detect this crossover and detect the gene order and break and rejoin the chromosome arms. So if I started here and numbered the genes 1, 2, 3, 4, 5, 6 down to 50, and this break is between gene 20 and 21, then this break is between 20 and 21. It's an exact exchange. But it's produced something new called recombination. Before, all we had was a complete blue strand or a complete purple strand, the same as were present in the parents of the individual producing the gamete. But now we do still have purple strands and blue strands, but now we have this chromosome that I will call recombinant. It's purple down to here, but then it's blue down to the end. This recombination means a novel combination of the genes on this number 2 chromosome, something that was not present in this individual or in that individual's parents. So given that these crossovers can involve any of these arms and can occur at any location up and down the arm, so you look from cell to cell, this one, the crossover occurs right where the pointer is, but in the next one, there may be a crossover in this location with this arm, and so on. It produces an uncountable and not infinite, but nearly infinite number of results in terms of allele combinations. I want you to remember this about sexual reproduction, our only way of reproducing. The reason sex is so successful is it produces huge diversity, huge variety. Now we can kind of see the power of the genome when we look at monozygotic twins. If you have a fertilized egg that divides and those two cells implant separately, you got genetic identity, and those two identical twins are remarkably similar in appearance. So the genes do make the same body. However, when you look at closely related individuals, and brother, sister are as close as you can get, the siblings in a family, people who have the same two parents, all have distinguishing features so that they each appear to be unique. And that's because of the power of sexual reproduction, meiosis and fertilization, in generating new combinations of alleles in every offspring. Now here is a different type of inheritance, and one that took us a long time to understand. It's based on that unique situation we have with the X and the Y chromosome in the male. The X chromosome is a large chromosome with lots of genes on it. The Y chromosome is much smaller, as we saw in the karyotype. That much smaller chromosome carries very few genes that are on the X, and many genes on the X are simply not present on the Y. So here we have the female. Now the female is the heterozygote in this case. You can see the big C and little c. And she is a normal color vision individual. Whether you're X with big C, or you're X with big C, little c, you have one dominant allele. So this individual, the mother, will be able to distinguish red from green when we're talking about red-green color blindness. Now here is a male. The male only has one X chromosome, so it makes 50% of its sperm carries the X with a big C, and this male has normal red-green vision. The Y, 50% of the sperm carry Y, and so have no allele for color vision. So when you copy cross the normal color vision male with the normal color vision female who is a carrier, you have 4 possible combinations. This individual does not express, the normal female in the upper left, does not express color blindness, and in addition to that, does not carry the little c allele, despite the fact that her mother did. Over here, we have the second kind of female who got the little c from her mother and therefore is a carrier, but because her father had normal color vision, his X chromosome has the dominant allele, and she is a heterozygote with normal color vision, but also who is a carrier. The bottom row shows the male offspring, one Y chromosome with the X big C would produce a male with normal vision, just like the father. And the second combination, the X with little c from the mother, will produce a colorblind male. Now, there's several interesting things about this that it takes a while to notice every one of them, but let's just point a couple of them out. If you have a female who is a carrier, then 50% of her sons, half of her sons, are expected to be colorblind. If you have a female who is a carrier of color blindness, none of her daughters will be expected to be colorblind, but half of them will be carriers. A male with normal color vision cannot have a daughter who is colorblind because his X chromosome makes that fertilized egg a female. A male with normal color vision cannot contribute any allele for color blindness to any offspring. It depends in this case on the X chromosome coming from the female. This condition here also shows the reason for a pattern in X-linked traits, which they call skip a generation. Skip a generation. So from this female, she may have had a mother who was a carrier or a father who was colorblind. Let's propose that her father was colorblind and that this X chromosome came from her father. So a colorblind father gives his allele to a daughter who has normal vision, but then that daughter who has normal vision, half of her sons, the grandson of the initial colorblind father, will show colorblindness. So colorblind to normal to colorblind, male to male transmission is common, not universal, but common in X-linked traits. Now, inheritance has taken a huge turn in recent decades because of DNA technologies. We now understand the DNA molecule and its code. We understand that its job is to produce proteins, and it is those proteins that are affecting the traits that we call inheritance. The ability to sequence, identify all the genes of the human genome and sequence them has allowed us to build a huge understanding of our DNA and protein variability. This was successfully completed decades ago, and we are now looking at the variability that's present in human genes. When we talk about sequencing the gene and identifying where it is on the chromosome, we are talking about mapping. The potential value of this is basically unlimited. We have united the karyotype, the chromosome with a map which contains the street address, basically, of every gene required to make a human, and every gene that varies that human condition. So for viable individuals, there are some traits that are pretty universal. One head is a good example. We have one head. If we have a embryo that starts to develop two heads, it doesn't appear as a live birth. Bilateral symmetry, bipedal locomotion. But there's other traits that just vary wildly, like hair color, all the way from almost translucent white all the way to blackest black, with red and brown variations due to accessory genes and polygenic inheritance. So we are cataloging this with a future view of being able to provide gene therapies. If a condition depends on the loss of a particular protein, then we can produce that protein in a test tube. A great example of that is the way we are now generating insulin from human genes, but outside the human body. This very busy figure, I mean, if you actually begin to think of the impact, if you actually begin to think of the importance of this DNA to protein transition, how it affects our viability, our fertility, and the various traits that we identify as disease traits or as problems in human development or challenges to human health, then you come up with this kind of desire to look at the human chromosomes, and here we've arranged them in order based on size, 1 through 22, and X and Y, along with the chromosome bands that were developed in karyotyping to indicate the major regions of the chromosome. And the importance of this map is that it is focusing on disease conditions and the genetic mutations that cause them. So just to start here, at the top of the right side, muscular dystrophy was talked about in chapter 10. And in position right here on the X chromosome. Prostate cancer, one of the so-called oncogenes, would be located on the number 1 chromosome right here. And Gaucher's disease, which is a storage disease in lysosomes, caused by a problem in glycolipid production in the membranes, is located here. So if you are a person studying genetic diseases and their frequencies in society, the Human Genome Project makes this kind of mapping possible. Many of these may be new to you, but I'm sure that many of them are also familiar. You've heard of Huntington's disease in this class, and we have discussed briefly cystic fibrosis. Here is diabetes mellitus. Chapter 18 will deal with that next semester. Type 1 diabetes is located right down here at the terminus of the long arm of the number 10 chromosome pair. Diabetes being an emerging and emerged epidemic which you will face throughout your medical career as a complicating factor in every parent's health. Here is something that's more difficult to find: Alzheimer's disease, which we recognize as a late life degeneration of the central nervous system and the brain. And it is located right here on chromosome 14. Breast cancer, the oncogene, or one of the oncogenes that makes breast lobes or ducts susceptible to a transformation from normal growth with contact inhibition to unregulated growth, producing a tumor in the human breast. So Down syndrome has a gene right here on the end of the 21 chromosome that affects its division in meiosis. So the presence in this element, the telomere of the 21, is what actually produces an egg typically that is unreduced. It has two number 21s. So the sperm carrying a number 21 gives you 3, and that unusual condition called Down syndrome. An individual with 47 chromosomes, we don't find many that survive with that number. But this is part of this genetic mapping activity. And that concludes chapter 29. So I got to do my business here, stop the recording. This is my first job.